Hello, everyone, and welcome to today's broadcast, Development of a Semi-Automated Closed CAR-T Manufacturing Process, presented by Dr. Calley Hirsch, a Development Engineer Scientist 2 from CCRM. I'm Dr. Susie Valdez of Labroots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by Labroots. Labroots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on that Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appear on that screen. Our speaker will respond to your questions via email. If you have any trouble seeing or hearing this presentation, just click on the Ask a Question box and let us know you're having a problem. This presentation is educational and offers continuing education credits. Click on the Continuing Education Credits tab located on the far right-hand corner of your presentation window and follow the process for obtaining your credits. Without further ado, please join me in welcoming our presenter today, Dr. Calley Hirsch. I will now turn the presentation over to her. Welcome, Dr. Hirsch.
Thank you, Susie, and thank you to Labroots today for the opportunity to speak about some of the work that we've been doing at CCRM to develop a semi-automated closed CAR-T manufacturing process. To start out, I just wanted to highlight a little bit about CCRM, which stands for the Center for Commercialization of Regenerative Medicine. This is a nonprofit company that was founded in 2011 with a focus to identify and commercialize new discoveries and technologies. We're located in Toronto's Discovery District in the heart of downtown Toronto. We've now grown to over 80 employees, continuing to specialize in this regenerative medicine field, and have expanded to also take on cell and gene therapy applications. We have a large degree of expertise in cell biology, as well as process development. We have specific interest in scaling up a number of therapeutic processes, and have recently entered into the GMP manufacturing area. We also have regulatory services, IP diligence and strategy, which is also associated with business development and strategy. And our focus is always innately focused on commercialization aspects, and more recently on company creation and incubating smaller companies. So as a whole, CCRM specializes in taking new concepts and technologies all the way through to the market and onto approval stage. We can work on helping build companies through incubations. So this is investigating regulatory, IP, commercialization potential, as well as technology. We have high technology expertise to be able to identify these companies that we feel will have success moving forward. We have an advanced division shown in the dark purple here. This is actually a research-based group, a wet lab group, and they'll take these early company ideas, perform proof of concept studies, and look to see how easily and readily these technologies may be translatable and commercialized. And more specifically in this group, we have a high degree of expertise in cellular reprogramming, as well as gene editing. Now, moving on through this process, after proof of concept discovery, we have another group called Bridge Division, which I'm part of. This is actually a collaboration between GE Healthcare and the Federal FedDev Ontario. And here we take discoveries that have been shown to work well in the lab, and we decide whether we determine and figure out how to scale these so that we can advance some of the manufacturing processes that are up there, optimize them, reduce costs, and so on. And now we have a new division called the Deliver Division at CCRM, and this is our GMP facility, which is opening in October of 2018, where we're going to specialize in phase 1-2 clinical trial production. We have 10 suites here, 14,000 square feet, and there are 2 suites that are dedicated to viral production, and 8 suites here dedicated to cell and gene therapy manufacturing, with over 25 experienced GMP staff members. So going back to the Bridge Division, we, again, are comprised of both CCRM and GE employees, and the beauty of this collaboration is that we actually coexist on the same floor and work seamlessly together day in and day out to achieve and advance different processes. So we offer process development solutions, optimization strategies. We look at strategies that will allow the greatest flexibility for our clients, as well as the industry moving forward, as well as have a great interest in scalable solutions across the cell and gene therapy field. We're also the process development arm for GE Cell Therapy Enterprises Solutions. And as some of our notable achievements, we have shown and demonstrated scale-up efforts for pluripotent stem cells, where we can achieve up to 36 billion cells in suspension culture at the 10-liter scale. Viral production is another one of our strengths. So we have suspension viral production systems, serum-free. We've shown scalability in these systems and the ability to also execute on downstream processing of this viral material. And we have emerging area of assay development that's quickly becoming one of our strengths. What I'm going to talk to you today about, though, is our expertise in cellular immunotherapies. So the reason that we're so interested in cellular immunotherapies is that these T-cell therapies are quickly taking off and showing extremely great promise in the clinic. There's over 150 companies that are pursuing T-cell immunotherapies, and a bunch of these are partnered with academic and research institutes, with some academics going their own route to put these products through the pipeline and into the clinic. So there's over 480 T-cell immunotherapy products currently in development. Of these, the most widely studied and tested are CAR T cells at 75%, TCR products, as well as TILs. So just very briefly, TILs take our tumor-infiltrating lymphocytes, take tumors out of patients. What they do is they grasp the tumor, specifically the lymphocytes that are associated within this tumor population, and they select for these lymphocytes, perform expansion of these cells outside of the patient's body, and then infuse them back into the patient with cytokine IL-2 in order to treat the tumor and to regress its progression. And that's just shown on the top here by this patient-based diagram. So CAR T therapies and TCR therapies are a little bit different. T cells are collected, or leukocytes are collected from the patient, T cells isolated, and then ultimately these T cells from the patient are genetically modified with synthetic receptors, either CAR receptors or TCR receptors. And these genetically modified T cells are then expanded further and infused back into the patient. And these T cells are unique in that they can now target the tumor cells specifically to be destroyed. And across the T-cell immunotherapy landscape and the ones in clinical development, the bulk of these therapies are largely associated with B-cell tumors. ALL, non-Hodgkin's lymphoma are the highest of those. And the the synthetic receptors themselves, chimeric antigen receptors, this is what the CAR stands for, are comprised of a number of domains. So again, these are synthetic receptors which contain an extracellular antigen binding domain, or a short chain variable fragment that specifically targets the cancer cell. This is shown as blue here in your diagram. And many of the CAR T therapies that are out there now and those that have been approved on the market are targeting CD19 and B-cell tumors. There's also a transmembrane domain that anchors the CAR to the T cell, and this is this next domain shown just downstream in the blue here, as well as different co-stimulatory molecules and activation domains that help to facilitate T-cell signaling to enhance the cytotoxic effect of the CAR T-cells and lead to tumor death. So as I mentioned, there are now CAR T therapies that have been approved for clinical use. It was exciting in 2017 to see the approval of Kymriah, which is used to treat in clinical trials, which showed 82.5% remission rates in relapsed refractory pediatric B-cell ALL patients. This was observed within 3 months of treatment. There's been approvals that have been expanded for Kymriah now in Europe and in Health Canada, and as well the indication ALL has been added as a target for Kymriah, both in the US, Europe, and Canada. Beyond this, the second CAR T therapy that was approved is named Yescarta. It demonstrated again in clinical therapies 51% clinical remission in DLBCL patients, and has also shown more recent approvals in Europe moving forward. So while CAR T therapies are demonstrating tremendous promise in the clinic, there's a lot of excitement surrounding whether or not these can be the next generation therapies for cancers. There are still a lot of challenges that we need to overcome. Specifically, targeting the tumors can be challenging. Hematological malignancies, we've now shown some success, but solid tumors, finding antigens that are specific to the tumor and not healthy cells has been a challenge. There's also side effects that have been associated with using these CAR T cells. So cytokine response syndrome has been shown in these patients, neurotoxicity, T-cell lysis, B-cell aplasia. So strategies to combat these are continuing to be studied. There's also a lack of control currently for the approved therapies, the ability to control, I should say, CAR expression. So different groups are now investigating how they can turn on and off this CAR expression in order to prevent some of the side effects that occur from CAR T treatment. However, I think one of the major obstacles that CAR T therapies need to overcome is this high cost that's associated with them. And in the upper left-hand corner, you'll see that Kymriah is priced at $475,000 US for treatment of ALL, whereas DLBCL is at $373,000, with a slightly lower cost being attributed to the fact that every patient treated will need to pay this price, and the higher cost being that only those that respond will be paying. Yescarta is similarly priced at $373,000 US. However, more recently in Britain, they've come back and said that they will not be refunding patients who receive these treatment because the price is just simply too high. So some of the things that contribute to these high costs are the complexity in the supply chain logistics. Once blood is taken out of the patient, it needs to be shipped in a cold environment, transported to a manufacturing site where T cells can be selected, expanded, modified, so on, and then eventually transferred back into the patient. And this requires another shipment step. But the complex manufacturing process also contributes to these high costs. And I'll get into the details of just how complex this process is moving forward. But the process in general is lengthy. It's 17 to 22 days from the beginning to the end of the process. So that's from taking these T cells out of the patient to putting them back. It's largely manual and it's open. So this means that it requires a lot of hands-on attention from lab personnel. It happens in biosafety cabinets, and it's open to sources of contamination. This also increases the variability of the CAR T products. When you have different hands on the product, you don't always get the same output. And it has been difficult to scale these processes, especially with a large number of unit operations occurring in a BSC hood. So some strategies to deal with this complex manufacturing process have focused on developing instrumentation for which you can take patient cells, put them in a box at the beginning, and at the end, the transduced patient cells will come out. But some of the issues with using these sort of systems have to deal with the fact that you're locking up this whole instrument for one patient at one time. So it's difficult to perform concurrent CAR T manufacturing processes. So one of our goals at CCRM was to look at developing a CAR T process that was kind of a next generation CAR T process. We wanted to systematically identify the unit operations throughout the manufacturing process, identify how we could make it more efficient, we could automate it, we could close it. And specifically, we wanted to identify and apply commercially available reagents that would be associated with enhancing this efficiency, and identifying automation strategies that were currently on the market to reduce these manual and open operations that were contributing to process variability, as well as potential contamination. And the last thing we were really quite focused on was the ability to maintain the process in a flexible manner. So we wanted to look at modular automated units that would allow for multiple CAR T products to be manufactured concurrently. So to start looking at the CAR-T process, we began at the beginning. And on the far left here is an outline of this manufacturing process. So very briefly, you take a frozen leukopak that you get from a patient. It gets transferred to the manufacturing site. At the site, the leukopak is thawed. These thawed cells are then subject to a PBMC isolation, which generally involves a wash to remove the lyse red blood cells. These PBMCs are then subject to a T-cell selection strategy. The T-cells are activated. They're put through an expansion. This may be a short expansion period, and then they're transduced with a chimeric antigen receptor. After transduction, the cells are further expanded. They're then subject to a harvest at the end of the process. They're washed to remove the media and any other contaminants. They're formulated in a cryopreservation buffer and then cryopreserved and shipped back to the patient. So to tackle this CAR-T process, to begin looking at automation, we first started by assessing whether or not we could automate thaw of this leukopak and the PBMC isolation step. So the experiments we designed for this are shown just over from the left. We chose to compare a manual method to an automated method. In the manual method, we took a frozen leukopak, and traditionally, as it had been done, we thawed it in a water bath. Following the thaw, this thawed leukopak was transferred to a biosafety cabinet. The cells were transferred into a bottle, centrifuged, washed multiple times, and then subject to a T-cell isolation. And this T-cell isolation was manual as well for the automated step. For the automated method, we took this frozen leukopak, and instead of using a water bath, we did the thaw in an instrument called the SmartMax. We then took this thawed leukopak and we put it on an instrument called the CPAX, where the cells were transferred into the instrument, washed to remove the lyse blood cells, and then the product from the CPAX was subject to a manual T-cell isolation. So the results from this work indicate that both PBMC recovery and viability are consistent coming out of the CPAX, which was nice to see. In addition, when we did this downstream T-cell selection, so that would be to get CD3 positive cells, we saw that CD3 positive cell recovery, viability, and purity were all preserved using the automated method versus the manual. So this indicated that us swapping away from the manual procedure of the water bath, BSC, and centrifuge to use the SmartMax and CPAX in its place was a viable strategy for our automated CAR-T process. Moving on now to the T-cell selection, what we did was we wanted to compare a number of T-cell selection reagents. We used a positive selection method where we would isolate CD3 positive cells. We used two of these strategies with the EasySep method or another CD3 positive method. And we also used a depletion strategy where we took cells that weren't bound to CD14 or 19 and assessed how these cells would perform. So for these experiments, we used these automated strategies that we identified in the previous slide. We took the frozen leukopak, thawed them on the SmartMax, isolated PBMCs on the CPAX2, and then split the product from these PBMCs into the different T-cell isolation methods. Following isolation, these cells were activated with immunocult activators. So these are CD3, CD28, CD2 T-cell activators, and all cultures were expanded in a similar manner. What we identified was that using the EasySep isolation to get CD3 positive cells, that we had the highest percentage of CD3 positive cell recovery or T-cell recovery, and that the purity remained high when we used CD3 isolation versus that of a depletion strategy. And this is not surprising that the purity would be higher in this case. We also looked at how the cells expanded after 5 days. So this would be 5 days post T-cell isolation or selection, and demonstrated that there was really no significant change in T-cell viability 5 days post-expansion, and that the expansion rates were similar regardless of the T-cell selection strategy. So given that the EasySep isolation method demonstrated highest CD3 recoveries and had high purity as well as high viability and maintained nice fold expansion throughout, we slotted this EasySep CD3 isolation step into our T-cell isolation method. So now pairing this T-cell isolation method with transduction step, which follows downstream in this workflow, we wanted to see how we wanted to see if the CD3 EasySep isolation method was also compatible with efficiently transducing CAR T cells. So again, we took PBMCs that had been isolated on the CPAX2. We split them into different T cell selection strategies. So the EasySep CD3 positive selection, a secondary CD3 positive selection, and a CD14, CD19 depletion strategy. Activated them with immunocult, expanded them, and transduced these cells on either day 0, day 1, or day 2. So this was also giving us another layer of investigation to determine whether or not these cells would be more effectively transduced earlier in the process, or if they benefited from later on. So just like in the previous slide, we identified that cells isolated using the EasySep selection strategy had amongst the highest level of transduction efficiency. And when we looked at how many days post-activation these cells needed to be, or how many days post-activation was suitable for transduction, and we identified that 1 day post-activation was most ideal. So now we've identified the CD3 EasySep selection method, fits nicely into the process. We're using the immunocult activators, and we've identified that transduction should occur on day 1 of the process. Next, we wanted to look into how the T cells were going to expand that we were isolating using these more automated and closed strategies. We elected to use a media called Zero-EM media, and we supplemented this with 5% human serum. So for these experiments, what we did was we took 5 different donors, and we wanted to investigate how well they grew over an 8-day period. Split into this, we also wanted to look at how T cells would grow in a bioreactor platform, or the Zuri W25 platform. So the outline of these experiments is shown more or less in the middle here. This frozen leukopak was subject to a smart match for thaw, PBMCs isolated on the CPAX again, CD3 positive cells isolated using the EasySep and activated with immunocult activators. Then these cells were grown in Zuri media on static flasks from day 0 to day 5. And on day 5, they were transferred to a Zuri bioreactor until day 8 of the process. And as you can see across 5 donors, we get comparable fold expansions that occur. Around 200 fold expansion occurs for each of these T cell donors. And we see that the overall average theoretical expansion being, 173 plus or minus 30. Across the process, cell viability remains above 88%, and viability is high on harvest day of day 8 at 97%. So using the Zuri media, as well as coupling the bioreactor into this process, demonstrates that we can achieve high degrees of expansion with high viability at the end of our CAR-T process. So while we liked how this expansion profile looked, one of the things that continued to bother us was that we were using static T flasks for our early expansion phase, and we wanted to see whether or not we could find a strategy to replace this. So what we did was we came up with this early closed culture alternative, and we assessed now how T flask expansion compared to this closed culture alternative. So T cells that have been isolated with EasySep, CD3 positive method, and activated with immunocult activators were either plated on T flasks for 5 days or in this closed culture alternative. The relative fold expansions were compared for these different strategies, and what we identified was that they were more or less similar regardless of whether T cells were grown in a T flask or this closed culture alternative. Cell viability was consistent across both of these culture platforms, and at the end of day 5, we had a similar percentage of CD3 positive cells, and the T cell subpopulations of CD4 and CD8 didn't seem to suffer or change much when T cells were grown in this closed culture alternative compared to the T flask. So this has now allowed us to identify an early closed culture expansion method and to get rid of the T flasks in our CAR-T process. Next thing that another unit operation that had remained open was our transduction step. So to look at how we could close this transduction step and automate it, we assessed an automated closed transduction method. So for this, we used the same unit operations that we had previously used, all the way up to EasySep, CD3 positive selection and activation using immunocult activators. The cells were plated in Zuri-M media on T flasks for 1 day, after which they were subject to different transduction methods. So in this case, on the far left in gray, transduction performed on day 1 was done in a T flask. So this was really a static transduction that occurred. We also transduced cells in this closed culture alternative to see how using this would impact the transduction efficiency of our T cells. We used retronectin, as shown in purple here, and retronectin is commonly used to improve transduction efficiency, so we wanted to see how that would compare to our automated closed transduction method. And then we used, on the far right here, this automated method to assess transduction efficiency. And for each of these methods, they were either expanded further on plates or remained in the closed culture alternative. And in the case of the automated transduction approach, following transduction, we transferred these cells to this closed culture alternative. And for all of these studies, we used a multiplicity of infection of 2.5. We've now tested 2 donors. We've looked at viability for these cells, either on day 4 or day 5 post-activation. We can see that viability is more or less consistent for all transduction methods. But interestingly, this automated closed method, either performs very nicely comparable to the other transduction strategies we assessed, or in donor 5 even was superior in its ability to transduce T cells. So coupling these observations now with what we've shown above for different automation and closure strategies, we're now able to close the transduction stage of the CAR-T process. Moving on, further down the list of unit operations in the CAR-T manufacturing workflow, we now hit and look to investigate this harvest and wash step, as well as the formulation and cryopreservation that we would need, steps that we would need to perform to generate our CAR-T product. 2 instruments were identified for this, the Cephia S2000 and the BioFreeze. So we took cells that had been expanded in Zuri-M media and on the Zuri bioreactor for up to 10 days. These cells were then collected on the Cephia, washed on the Cephia, formulated, in this case on the SmartMax for the experiment, and then ultimately frozen in the BioFreeze. Looking at this workflow, we see that viability of T cells pre-Cephia and post-Cephia is high, well above 95%. Recovery, T cell recovery post-Cephia ranges around 90% or 86. And viability, once we now thaw this CAR-T product, was high as well, near 100%. So we also saw relatively high post-T cell recovery, indicating that we weren't losing a lot of T cell product by implementing these automation steps. So both the Cephia S2000 and BioFreeze were slotted into our CAR-T process. So just as a summary for what I've shown so far, we've gone through all the unit operations. We've identified automated closed solutions for the bulk of these steps. Briefly, we're using the SmartMax to thaw the leukopak, CPAX2 to isolate PBMCs. This EasySep CD3 positive step, unfortunately, still remains open, and we're using this for T cell selection. We transfer cells to a closed culture alternative. We activate with immunocult activators, expand cells in Zuri-M media. Transduction is performed in an automated closed by an automated closed transduction method. Cells are further expanded in this closed culture device for 2 days post transduction, and then they're transferred to a bioreactor for further expansion, where we have perfusion strategies and different set points that are maintained in order to allow for efficient T cell expansion. At the end of the culture, we're going to be shooting for day 8, but the further results I'll show you are at day 10. We harvest cells using the Cephia S2000 and subsequently cryoformulate in the S2000 and then later freeze this CAR-T product in the BioFreeze. So we're at the stage now where we have performed some simulated CAR-T runs. We've done this using GFP virus, and I'll just show you where we're at in regard to that. So the first part of the CAR-T process, the PBMC isolation and T cell selection, is shown here on this first slide, where the leukopak is thawed in the SmartMax. PBMCs are isolated in the CPAX, and then we have an open CD3 selection process that is occurring using a magnet-based approach here. The results from the two donors are shown below. We see relatively high PBMC recovery following the cells coming out of the CPACs. We also see that viability is high. It's maintained above 95%. And that CD3 positive cell recovery is in line to what we've seen previously. Greater than 66% of CD3 positive cells are recovered. These cells are 95% pure and remain highly viable. Now, post CD3 selection, the cells are activated with immunocalled activators, and they're expanded further in a closed culture alternative for 1 day, after which they're subject to transduction using this automated closed transduction method. Following transduction, again, they are transferred to the closed culture alternative again, and then ultimately sent to the Zuri for this final expansion phase. What we see when we look at theoretical fold expansion across these two donors that we've looked at is that we can achieve 10 or 10 billion transduced T cells on day 8, which is pretty exciting. We transfer the cells to the Zuri on day 4, at which point we see a really nice increase in the expansion rates, and viability remains high across the process. We also have noticed a nice level of transduction efficiency using this automated closed transduction method, where at day 10 of the process, we have approximately 80% of T cells that remain transduced. And when we go on to look at the phenotype of cells that are harvested, and these results actually shown from day 8, but day 10 is similar, we see that there's 30% of the population that remains CD8 positive, nearly 60%, 66% CD4 positive, and then looking at CD4 and CD8 phenotypes further, a large percentage of these cells reside in the central memory phenotype, with the CD8 population containing a number of naive cells as well. So all in all, when we look at the process, we're able to achieve 10 billion transduced T cells on day 8 when we start with 200,000 T cells. Sorry, pardon me, 200 million T cells. Now moving on to the harvest stage, so using a Cephea S2000 to harvest, wash, and cryoformulate, followed by the preservation and the biofreeze, we see that pre-Cephea cell viability is high. This is maintained post-Cephea as well. Recovery following the Cephea also remains high in 2 donors, which is nice. One area that we are continuing to work on is that recovery post-thaw drops off once we take these cells out of the liquid nitrogen tanks to see how they recover. Nicely, we also demonstrate that cells remain a high degree of GFP expression, with over 80% of cells transduced once we recover cells out of this cryopreserved state. So what I've discussed with you today is that we're working on the development of a semi-automated closed CAR-T manufacturing process. We feel that this process can be widely adopted across the T-cell immunotherapy industry. We achieve 10 billion GFP transduced T cells in an 8-day process, and we've now shown this with 2 donors. We're using a number of commercially available reagents that includes the EasySep release human CD3 positive selection kit, as well as immunocalled human CD3, CD28, CD2 T cell activators, and Zuri T cell expansion media supplemented with human serum. Beyond this, we've identified automated integration strategies to remove some of these manual and open operations. This also leads to a large reduction in the labor requirements for the process. So outlined here are these instruments that I've discussed with you throughout. And while identifying these reagents and automated strategies, we've been able to maintain flexibility of the process so that we're not tying up any of the instruments for prolonged periods of time to prevent further CAR-T processes or products from being concurrently manufactured. So as for next steps, we're looking to complete more simulated CAR-T manufacturing runs. We're moving on to perform a minimum of 3 CAR-T engineering runs, where we'll also assess the cytotoxic activity of our CAR-T products. Really interested in identifying a closed T-cell selection operation, or even finding a way to close this CD3 EasySep isolation method. Now we're also investigating strategies by which to improve our Tcell post-thaw recoveries. And with that, I just want to wrap up by thanking a number of individuals who have worked on this project, both CCRM and GE Healthcare employees, as well as our funding from FedDev Ontario and the Government of Canada, which supports our Centre for Advanced Therapeutic Cell Technologies. Thank you.
And thank you, Dr. Hirsch, for that informative presentation. And we will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box and located on the far left of your presentation window and type your questions into the box that appear on the screen. Click the Send button. Our speakers will follow up with you via email. So let's take a look at some of our viewers' questions. Dr. Hirsch, are the reagents described in your presentation GMP compatible?
Yes. For the most part, all of the reagents that I've discussed today are either GMP compliant now, or they're in the process of being, or they're in a pipeline to be designed to be GMP compliant and available within the next year or so. As well, the automated strategies that we used are all readily being utilized in different manufacturing processes and can be readily implemented immediately. So that's pretty exciting.
And Dr. Hirsch, how will the process change as manufacturers aim to reduce manufacturing timelines?
There's a really big interest in reducing the timelines for manufacturing. There's reports that have shown that using cells that have expanded for less time, have greater efficacy in patients. And I think the ways that we're going to be able to achieve this is by identifying specific T-cell phenotypes that create the best effect in patients. So if we can identify these phenotypes, isolate them effectively, and then ultimately use this smaller population of more potent cells to treat the patients, I feel that it will not only combat some of the side effects, but it will also reduce these manufacturing timelines and costs that are associated with the CAR-T process.
Perfect. Thank you, Dr. Hirsch. And one final question. How will alternate or non-viral genetic modification strategies influence the CAR-T process?
So there's a great interest in identifying different non-viral strategies. Virus production is extremely expensive, and it's a lot of the cost contribution to what we're seeing here for these CAR-T products. The issue with these non-viral delivery strategies is that many of them are not efficient, so we have to get the efficiency levels up. But there's also other methods, such as using transposons, to create CAR-T cells that are readily being used and showing promise. So I think this is going to become, a really fast-paced new area of interest moving forward.
Thank you, and thank you for your presentation today. Do you have any closing remarks you'd like to share with our viewers? I just wanted to say, I guess, that the process that we're developing, is very much still in the development stage. We're working to completely automate this process and close it. And I hope that everyone just enjoys the update that I was able to provide today. Thank you. And thank you again for your presentation and for your important research. I'd also like to thank Labroots for making today's educational webcast possible. Before we go, I want to remind everyone that today's webcast will be available for on-demand through December 2018. Please share that email from Labroots letting you know that the webcast will be available for replay. And please share that announcement with your colleagues who may have missed today's event. That's all for now, and thanks for joining us. We hope to see you again soon. Bye-bye.